Welcome back. Next we'll move on to chapter number 10, blood, and various components found within blood. Now, blood is a mixture of various types of cells and fragments of cells, dissolved chemicals, hormones, oxygen, waste, and so on, that go from to and from cells throughout the body. Now, blood is made up of two separate parts, a liquid part and a solid part. The liquid part is called plasma, and the solid part is the formed elements, or the cells, red blood cells, white blood cells, platelets. So if it's liquid, it's plasma. If it's solid, it's the formed elements. Now, in a normal, healthy person, these percentages of how much there is of each type remains fairly consistent. Plasma is about 55% of overall blood sample. The white blood cells and platelets are a little bit less than 1%. And then the red blood cells are 45%. Again, in a normal, healthy person. When these percentages get much higher or much lower, that indicates that something is wrong. And the percentage of the red blood cells in a given sample is called the hematocrit. So the hematocrit is a blood test. The percentage of red blood cells, which will be roughly 45% of a sample. So if it's too high, it's being made too much, which is a problem. If that's much lower than 45, then something is wrong. It's most likely an injury somewhere where you're just bleeding. Alright, here's all that just in one image. A typical blood sample. This yellowish part would be the plasma, a little more than half. Platelets and white blood cells, very, very small percentage. And the other 45% red blood cells. Now, all blood cells are formed within the red bone marrow of long bones called hematopoiesis. So, there's one initial stem cell that leads to more specific cells and more specialized cells, and then from that one stem cell, that's where you get all the white blood cells, all the platelets, all the red blood cells. So all blood cells are formed within bone marrow, and this is how this looks. And I would not put this on a test. It's more to show you how complex this process is. All blood cells start off here, the hemocytoblast. Then they further specialized into these two types of cells. And this type of cell will break down into red blood cells, platelets, and various forms of white blood cells. So this is how complex the system is. But all blood cells are made in the bone marrow through hematopoiesis, and they're made by this beginner stem cell here. Alright, first we'll talk about the red blood cells. The formal name for red blood cells is erythrocytes. The prefix eryth, E-R-Y-T-H, means red, so red cells, red blood cells. Now, these cells are very flat, biconcave, which means they cave in on two sides, and are very, very flexible. So this is how they're able to kind of wiggle their way in and out of blood vessels fairly easily. Now, this very unique shape allows them to have more surface area to carry more oxygen. That's their main role in your body, is to carry oxygen to cells. And this oxygen is carried through a pigment called hemoglobin. This is how one would look if you're looking at it from the top, kind of like a filled-in donut almost. If you look at it from the side, it caves in on this side, caves in on this side. That's why it's called biconcave, or caves in on two sides. Okay, if you're just zooming on one red blood cell here, this is how oxygen is actually carried in 4 units here. There. If you were to look at a blood sample on a slide, a great majority of the cells you would see are red blood cells. They would look like this. And you'll see these scattered white blood cell here and there. Alright, move on to white blood cells, also known as leukocytes. The prefix leuko means white. Sometimes you'll see this spelled L-E-U-C, and there's no difference, either the C or the K. Leukocyte, it still means the same thing, means white blood cell. And their main job is to fight infection. And depending on what type of infection that you have, will determine what kind of white blood cell is made in higher numbers. Now, there are 5 types of leukocytes, and they fall into 2 major groups: the granulocytes and agranulocytes. And now all these cells will vary by their overall size, the shape of the nucleus, the type of cytoplasm, and what color they appear on the slide. First we'll talk about the granulocytes of the white blood cells. And they're called granulocytes because their cytoplasm looks grainy. It looks like there are grains of sand within the cell. That's why they're called granulocytes. These only live for about 12 hours approximately. And some examples of granulocytes: neutrophils, eosinophils, and basophils. Neutrophils, their nucleus can have anywhere between 2 to 5 different lobes, or 2 to 5 different sections. These will engulf and phagocytize bacteria, fungi, and some smaller viruses. And this is a great majority of white blood cells. About 55% of all white blood cells are neutrophils. This is how one would look. You can actually see how it looks kind of gritty and grainy in the cytoplasm, which means it's a granulocyte. And this one, 2, probably about 3, maybe 4 lobes of the nucleus. Of course, the RBC, red blood cells, are all around it. Eosinophils. What is unique about these, when a blood sample is stained and put on a slide, these show up very, very dark red. One of the 2 blood cells we'll talk about that are given away by its color. So eosinophils stain a very dark red, almost like a brick red. Nucleus usually has only 2 lobes, and these will fight against parasitic infections. Now, these are not very common. These are less than 3% of all white blood cells. This is how these would look. Now, of course, the grainy cytoplasm, the granulocytes, 2-lobe nucleus, here, right, 1, 2, and it stains a much darker red compared to the stuff around. Basophils, this is the other type that's very identifiable by its color. These will stain a very dark blue color, almost like a purple color. These will migrate to tissue that's been damaged and release histamine. This will promote inflammation and allergic reactions. Those of you who have allergies, this is why you take antihistamines, is because of this compound right here. This is why you have the dry eyes, or the itchy eyes, or the dry throat, or the runny nose. It's because of this chemical being produced by these cells. Now, these are not very common at all. These are less than 1% of all white blood cells. And again, you can see the grains of sand inside, or appears to be grains of sand, the granulocytes, very dark blue, almost purplish color. So this is the cell that releases histamine. Alright, next we'll move on to the second class of white blood cells, the A granulocytes. And they're called A granulocytes because they do not have that grainy appearance. The prefix A or an means without. So without grains, without granulocytes. Both of these tend to be much larger than the cells around them. They usually have one large, non-segmented nucleus. And there are 2 types of these, and they're very common to confuse with each other, but I'll give you a way to keep them straight. The 2 types of A granulocytes are the monocytes and lymphocytes. Monocytes, these are the largest of all blood cells. These are about 3 to 4 times the size of a typical red blood cell around it. So the size difference is very, very noticeable, and you'll see it when I put up images here in a second. But there's a very large size difference between a monocyte and everything else around it. Now, after a few days of monocytes being produced, they will leave the blood and enter tissues to help fight disease. When they do this, once they enter the tissues, they're now called macrophages. And from there is where they'll digest bacteria, other dead cells, other debris from cells. And these are about anywhere between 5 to 9% of all leukocytes. Again, all these around it are typical red blood cells. See, one monocyte. See how much bigger this one cell is compared to this. One large nucleus and no grainy appearance. So this is an A granulocyte. An MO for monocyte. The next lymphocyte, what gives this cell away is the very large nucleus, to the point where all you really see is a very large nucleus and a very thin ring of cytoplasm around it. And these are the cells that play a major role with your immune system, producing the B and T cells. And these account for anywhere between 25 to 33% of all white blood cells. And these are larger than the red blood cells around it, but not as large as the monocytes. Again, all these cells around it here are red blood cells. One lymphocyte, very, very large nucleus. It's usually much larger than this, but this one cell is still much bigger than these. Here they are side by side, monocyte and lymphocyte. Again, this is about twice the size of a red blood cell. Very large nucleus. Usually, only thing that's really visible is a very thin ring of cytoplasm. Compare that to that, which is big, but look at this compared to this. Much, much larger than the cells around it. In this image, this hasn't been altered at all, hasn't been photoshopped at all. The nucleus does kind of look like a heart in this image. So, monocyte, lymphocyte. Both of these are examples of A granulocytes. Here are all 5 of the ones that we just talked about, all on one image. The first 3, granulocytes, neutrophils, grainy appearance, multi-lobe nucleus. The eosinophils, the 2-lobe or bilobe nucleus, stains a very dark red and still grainy. And the basophil, grainy appearance, is stains a very dark blue. And the last 2 we just talked about, the A granulocytes, monocytes, much, much larger than everything else around it. And the lymphocyte, very, very large nucleus and not much else that you can see. Now the last type of cell we'll talk about are platelets. And their formal name is a thrombocyte. Now these lack a nucleus and technically are not complete cells at all. These are really fragments of a prior larger cell. And their main function is to form a thrombus or to form a clot to stop the bleeding of an injury. And these tend to last only about a week and a half, so no more than 10 days or so. But thrombocytes are not considered a full cell. They're just fragments of cell. Okay, on this image, the arrow is pointing to one platelet here, but also that is a platelet, that is a platelet, that. All these small bits of a cell, those are considered to be platelets. Okay, here's one image of a blood sample with everything that we've just talked about all on one. Neutrophil here, eosinophil with its red color, basophil with its dark blue, almost purplish color here, monocyte, large size difference, lymphocyte, and then that small piece right there, platelet. Okay, next we'll talk about the process of hemostasis. This is the process that will stop bleeding in injury. Now I have it boiled down into 3 major steps. There's actually a lot more that goes on to it than the 3 that I've given you, but I'm grouping 4 or 5 other steps into one of mine. So boil it down to just 3 key major steps. The first one is called the vascular spasm. It's the smooth muscle of the damaged vessel contracting to lessen the amount of blood that's lost. And think of this as a reflex. If you touch something that's very hot, you pull back so you don't get hurt anymore. It's the same thing here. When a blood vessel gets damaged, it will contract so it doesn't lose any more blood than it already is. So think of that as instead of a spasm, think of it as a reflex. The next step, platelet plug formation. This is where platelets will stick to each other and stick to the ends of the damaged vessel. This is the very beginning of the plug to help stopping the blood loss. Because if there's a hole there, that hole has to be filled. So it's initially filled with platelets. And step number 3, coagulation. This is the actual forming of the blood clot itself. There's a protein called fibrin that is created in very thin, very strong, very sticky threads. And these form basically a big spider web over the damaged area. And this will collect other cells as they go by, and this will help form a clot over that damaged area to help fill that space so less blood is lost. Okay, next we'll talk about 2 terms that are often confused with each other, thrombus and embolism. A thrombus is just a general term for a clot that forms abnormally, when a clot shouldn't be in a vessel. Now when a piece of that clot breaks off and starts to travel throughout the body, that's when it's called an embolism. So if it goes into the lungs, it's called a pulmonary embolism. This can be not only very painful, but can be fatal, depending on how large that piece of clot is. So a thrombus is just a term for a clot in general, but when the piece of that breaks off, it starts to float around in the body, that's when it's called an embolism. All right, here are those 3 steps all on one image. Here's the injury, this blood vessel here. The first step, the vascular spasm. So you're going to contract to lessen the amount of blood that could be lost. Step 2, that platelet plug formation. You get platelets traveling to this area, sticking to the ends of the damaged tissue, and then also to each other to help block that space. And the third step, the actual formation of the clot, coagulation. That fibrin getting together, forming a interlocking web of sticky tissue that help trap other cells as they go by, forming a clot. This is how one actually would look. These are all red blood cells here. And these very thin, sticky threads, looks almost like a spider web, are the fibrin threads. And that's how it would look when it's completely filled in with fibrin. And these individual circles here are red blood cells, and everything else is fibrin, a big fibrin mesh. Okay, the last major topic of this chapter are the ABO blood types that we have. Now, there are only 4 types of blood that exist, either type A, type B, type O, or type AB. So you will have one of those 4. Now, of those 4, type O is the most common. Over, I think, 50% of all people in the world are type O. And the least common is type AB. Now, what determines a person's blood type is either the presence or absence of a certain molecule that's on the surface of red blood cells. Alright, for each blood type, they have their own specific antibodies, and antibodies are what will fight infection. And these are there to help prevent against any foreign blood. So if your body senses something that's in your blood that shouldn't be there, it's going to be attacked. So this is why you need to match blood types, very particularly. You can't just grab some random bag off a shelf, at a blood bank and give it to just anyone. This has to be matched correctly. If they're not matched, then there are some very serious complications. People who have type A blood, they have anti-B antibodies. If people have type B blood, they have anti-A antibodies. If people who have type AB blood have no antibodies. We'll talk about why that's important here in a second. And people who are type O, which is the majority of people, have anti-A and anti-B antibodies. And like I mentioned, if these blood types are not matched correctly, the red blood cells will start to fight each other, and they will start to clump together. This process is called agglutination. When these red cells are clumped together, they can't function. Then they eventually will die. And if red blood cells die, that means oxygen can't be delivered to tissues. That means tissues will die, which means organs will die, which means you will die. So this is why it's incredibly important to be able to match blood types correctly. And there are 2 particular names given to types of blood. For those of you who have type O blood, you are called the universal donors because you can give blood to anyone. Because, going back to here, no other blood type sees you as being foreign. Type A won't have a problem with you, and type B won't have a problem with you. And type AB blood can receive blood from anyone. They're called the universal recipients. So there's O in donor for type O. Be the best way to keep these straight. Type O can give to anybody, regardless of what type they have, and type AB blood can receive from anyone, regardless of what blood type they have. All due to the fact that they don't see other types of blood as being different or foreign. Here's a way to represent all of that in that process of agglutination. If you have type A blood, the presence on the surface here with the anti-B antibodies here. So if you're given something that your body doesn't recognize, it will fight it, clump the cells together, and eventually rip them apart to the point where they can't function because they're either clumped together or they're being torn in half and they can't function if they're not together. And this is how agglutination would look. Instead of having that characteristic biconcave, flat, disc-looking shape to them, these are all clumped together. And at this point, these cells are not going to be able to function. Alright, next we'll talk about the RH group. It's called the RH because it was first discovered in the rhesus monkey. So that's where the RH comes from. And all this is the positive or negative of your blood type. So if you're O positive or O negative, AB negative, AB positive, that positive and negative indicates whether or not you have a particular type of antigen on the surface of your red blood cells. If you have it, you're going to be positive. If you don't have it, you're going to be negative. And a great majority of people, over 80% of people, are positive. So if you're type O, odds are you're going to be O positive. If you're A, most likely you're going to be A positive. B, B positive. Or AB positive. Alright, that brings us to the end of Chapter 10 on blood. If you have any questions, please feel free to contact me.